Hi, this is Dr. Steven Feldman. I am professor of dermatology, pathology, and public health sciences at Wake Forest School of Medicine in Winston-Salem, North Carolina. This is the red line in which we will discuss how biosimilars have begun to impact clinical practice and patient access to treatment with a biologic. There are already a variety of FDA-approved biosimilar products. Filgrastim was approved in 2015. It's a biosimilar to Neupogen. Infliximab, biosimilar to Remicade, has been approved. The FDA has also approved etanercept and adalimumab biosimilars. These drugs are used for a wide variety of indications, particularly inflammatory diseases. The clinical impact of biosimilars, to me, is somewhat limited. There is the thinking that biosimilars will come in at lower costs, but it's not going to be dramatically lower cost. But that lower cost will often lead to lower cost of the reference biologic as well. There's some evidence that the development of biosimilars may lead to greater use of biologics. Now, if a patient is doing well on a reference product, that patient should also do well on the biosimilar. So there's not a lot of change there. If the patient is not doing well on the reference biologic, switching to the biosimilar is not likely to be of any benefit, because the drugs are basically the same thing. So biosimilars are not going to help a patient who is not doing well on a reference product. Now, there are several issues that we're going to want to discuss with regard to biosimilars, and that includes interchangeability, substitution, and pharmacovigilance. But as I consider these issues, I like to keep in mind that the biosimilar is basically, at least to my thinking, very much like another batch of the innovator product. And with that in mind, we can understand interchangeability, substitution, and pharmacovigilance, I think, much more clearly. The primary reason people are excited about similars is the potential that they will reduce the cost of therapy. And here we have total expenditures for filgrastim products. And you can see that the amount expended on the biosimilar gradually increased. The amount that was spent on the innovator product gradually decreased somewhat, and the total cost came down some. Now, that amount that it comes down appears not to be very large, but one of the neat things is we might have expected that over time the expenditures for the filgrastim products would have grown and might have grown considerably. And so one of the advantages of biosimilars is that even if they don't reduce the spending on biologics, they may at least prevent rapid growth in the spending on biologics. And that may be an enormous benefit. Extrapolation is of concern to some people. This is the idea that an innovator product is tested for efficacy and safety in a wide variety of conditions. The innovator biologic will be approved for multiple different indications based on multiple randomized controlled phase 3 trials. A biosimilar does not undergo multiple randomized controlled trials in different indications. It may only be tested in one indication. And if approved, its use may be extrapolated to other indications. The idea being that if you've shown the biosimilar is similar to the innovator product, it should be good in all the diseases in which the innovator is good. Biosimilars are allowed to be extrapolated based on a totality of evidence. If the totality of evidence, which includes the physicochemical assessment of the structure of the biosimilar and its functional characteristics, how well it binds its target, and its pharmacokinetic and pharmacodynamic characteristics, how well it stays in the blood, and how well it actually functions in a particular disease. If all of that shows similarity, then biosimilar would be allowed to be extrapolated for use in all the conditions that the innovator is indicated for. And basically, the ultimate decision about whether to use a biosimilar or the originator product would come down to other considerations, predominantly the cost of the product. There's concern about telling whether a biosimilar will create some problem. So there's attention paid to pharmacovigilance. The FDA may approve the biosimilar, and then once in use, you want to have data on the biosimilar to make sure it's performing as expected. And you can do this with registries and all. Now, some people feel the biosimilar should have to have a distinct name so that you can track it. And to me, the biosimilar is basically like another batch of the innovator, and we don't give each batch of the innovator a different name so that we can, make sure that it's working the same as the previous batch. To the extent that we do give a biosimilar a different name and carefully monitor it, we are actually already at a higher level, a higher bar for making sure that the use of the medication is safe and effective than we do for the different batches that we're currently very happy with. The efforts to identify risks may include healthcare provider communications, recalls, alerts, and potentially even REMS programs. Now, those are risk evaluation and mitigation strategies. As healthcare providers, we may have some role in pharmacovigilance. The FDA MedWatch program records adverse events that are reported. There is tremendous underreporting of adverse events. And so that program is only as useful as we make it. If we report adverse events when we see them, it will be more useful than if we don't. There's potential for medication errors that could be reported as well. If an adverse event occurs, it has to be attributed appropriately, and we want to know what the patient actually received. You'd have to know whether it was the innovator or the biosimilar, ideally what batch of the innovator or biosimilar was used. Here are some tips that have been recommended. When a patient is on a biologic, you want to be aware of whether they're on the innovator or which biosimilar that they were prescribed, which they were dispensed. You want to make sure you're using the right trade name that defines exactly which product they're on. You want to contribute to registries that will follow people over time, and it's one of the best ways of collecting efficacy and safety data and monitor for long-term safety data and encourage transparency in drug characterization. I think that those are all interesting and good tips, but here's the thing. We've been using different batches of innovator products for over a decade now, and we really haven't done any of these things except perhaps the issue of registries. And I'm still comfortable with my patient moving from one batch of an innovator product to another, and those minor variations that occur between batches are of no relevance to me. I think making sure that we get as many patients as possible into registries will be valuable, and not just because of the introduction of biosimilars, but because of the variation in the innovator products as well.
Now that we've learned about the implications of biosimilars on clinical practice, let me summarize. I think we can think about biosimilars as being just like another batch of the reference product, and that has important clinical implications. If a patient's doing well on a biologic, the biosimilar should work fine. If we're thinking about putting a patient on a biologic, putting them on the biosimilar should be fine. But if a patient's not doing well on a particular biologic, switching them to a biosimilar of that same biologic should not be expected to give them any benefit, because the biosimilar should act just the same way the innovator biologic does.